Good morning, everybody. You're all welcome to this morning's session. Sorry for being late a little, but it's all for towards having a very good seminar this morning. This morning, we would like to start with the opening prayer. And that will be done by Reverend Mrs. Eunice Ampim Danso.
Good morning. Thank you for having me. Shall we please bow our heads for a word of prayer? Bless the Lord, O my soul, and all that is within me, and all that is within us. All that is within Uguchi, bless his holy name. Bless the Lord, O my soul, and forget not all his benefit over the past 45 years, O Lord. Daddy, Lord, today another session, Father, of the lecture series is here. We ask that you will take preeminence over all proceedings over here, even in the name of Jesus. Daddy, Lord, my prayer is that at the end of this lecture today, I pray that there shall be new, Father, research ideas, new research strategies, Father, for a fruitful and profitable findings in the near future. Daddy, Lord, we bless you. We give you all the glory. As Prof. Koram starts with us this morning, let your awesome presence, Father, be with him throughout this study. And let there be an understanding, even in the name of Jesus, we have prayed with thanksgiving. And all shall say amen.
Mrs. Eunice Ampim Danso for such a powerful prayer. This morning, I would like to also invite our acting director, who is also a malaria researcher, to give us a quick word on behalf of the director. Prof. Ofori.
Thank you very much, Chantal. So on behalf of the director, who was actually looking forward for this day, the presenter is one of her mentors. And I also stand here as one of my great mentors. So she wants to welcome you all to this very special seminar on malaria. The control, elimination, and management of malaria cannot be written without Noguchi since its inception of the institute. So today we are very, very happy to have one of our greatest scientists in the area of malaria research to present something on behalf of the whole malaria group in the institute, which over the years we have done so well. So she, even though she's not here, she will be with us online. So surely he will be looking forward to listening to us. So I'm very happy that we have all turned out on our numbers. And as you all know, we are celebrating our 45 years since our establishment. And over the few weeks, we have been presenting some of the work that we have done that has had impacts in the health of many Ghanaians and also people across the globe. So today we are very happy to have you here. As we prepare to climax it in November, so there are a lot of activities as we also line up for this, and we are looking forward that everybody will plan his or her activities so that we all together we can celebrate the 45 years of our existence as an institute and the good work that we have done. So I won't say much, but I'm sure the director is also listening. So I'll hand over to the MC so that you introduce the chair for this morning's session. Thank you very much.
Thank you, Prof. Thank you. So I'll quickly proceed and introduce our chairperson for this morning. Our chairperson for this morning is a professor of epidemiology. He's a distinguished social epidemiologist with extensive training in both social science and epidemiology. His work blends epidemiological and implementation research with a focus on answering crucial questions that inform both national and global policy and practice. When it comes to his research interests, his interests span multiple disciplines, including social science, epidemiology, and implementation research, with special emphasis on diseases of public health importance, such as malaria, of course, filariasis, Buruli ulcer, TB, antimicrobial resistance. He also has expertise in clinical trials, adolescent reproductive health, and climate change. His work on malaria has focused on intervention implementation to reduce the burden of disease in the population. He has participated in clinical trials, conducted studies on social determinants of malaria infections, treatment, control, and implementation of malaria control interventions. One of his significant contributions is his work on intermittent preventive treatment for children, that is IPTC, which has helped guide global strategies for malaria control. When it comes to consultancy, advisory, and leadership roles, our chair for today has consulted for prestigious organizations such as the Ghana Health Service, the Ministry of Health, the WHO, the Gates Foundation, USAID, Johns Hopkins, Bloomberg School of Public Health, the Global Institute for Disease Elimination, and the Ghana Statistical Service. He has held a visiting professorship position at the New York University's Abu Dhabi campus and the University of Global Health Equity in Kigali, Rwanda. He currently serves as the vice chairman of the Ghana Schistosomiasis and Soil-Transmitted Helminthiasis Expert Committee, and is a member of the Anti-Malarial Medicine Policy Review Committee under the National Malaria Elimination Program. He is a member of the Scientific Advisory Committee for the Foundation for the National Institutes of Health in the United States. When it comes to professional affiliations and editorial work, our chair for today serves as an editorial board member for BMC Public Health and Health Sciences Investigations Journal. He is a member of several renowned academic and professional organizations, including, just to mention a few, International Society for Infectious Diseases, American Society for Tropical Medicine and Hygiene, International Society of Substance Use Professionals, and the Partnership for Social Sciences in Infectious Diseases of Poverty. I think I need to pause here. If this was the National Math and Science Quiz or the popular Noguchi Interdepartmental Quiz Competition, I would have said the last clue is, but today is not the quiz day, so but I'll still proceed and give the last clue for us to see if we will know who our chair for today is. He has served as the head of the epidemiology department since 2021, a role he has held from 2013 to 2017. I believe he may be the only head of department with 2 English names. Now, ladies and gentlemen, please join me in giving a warm round of applause as we welcome our distinguished chairperson for this morning's program, Professor Stephen Collins Aholu. Yeah, welcome, Prof, your audience.
Thank you very much, Dr. Agbemebiase, for that wonderful introduction. Sometimes when you hear this, you feel flattered, but to God be the glory. So we are happy to be here this morning. This is supposed to be the 4th in the series, but it turned out to be the 3rd of the 45th anniversary lecture series. And this is on malaria. And when I joined Nguchi, the whole institute was noted for malaria. So everywhere you go, everybody identifies you as those who work on malaria. before HIV became popular, there was malaria. Today, we have one of our own to give us today's lecture, somebody who have worked extensively on malaria research, somebody who all of us working on malaria have worked with in one time or the other. And he is a former director of the Institute, serving from 2012 to 2017. He's a professor of epidemiology with nearly 3 decades of research experience in malaria epidemiology and public health, including clinical trials of novel interventions against malaria. He established the program for therapeutic efficacy monitoring and drug resistance that has continued to provide evidence for drug treatment policy in Ghana. He served as a coordinating member of the Africa Malaria Vaccine Network, which later became Africa Malaria Vaccine Trust, under the able leadership of Professor Kilama of Tanzania. This group was instrumental in the establishment of research ethics committees and at several research institutes in Africa during the late 90s. He also served as a member of independent data monitoring committees for clinical trials in Africa, including the RTSS malaria vaccine developed by GSK. He currently leads the clinical trial unit at the Noguchi Memorial Institute for Medical Research and actively leading the institute in the conduct of early phase trials. He is the lead investigator for the COVID-19 research outcomes worldwide network for coronavirus mitigation trials. This is an international multi-site, adaptive, randomized, placebo-controlled trial assessing the effectiveness of candidates' interventions in mitigating COVID-19 disease in healthcare workers. He is also leading a phase 1B clinical trial to evaluate the safety, tolerability, and immunogenicity of a vaccine in a healthy population, healthy adult population. He has more than 120 peer-reviewed publications and teaches and supervises graduate students at the School of Public Health and the University of Ghana Medical School. Please kindly join me in welcome Professor Kwadwo Ansah Koram to the podium for today's lecture.
Thank you very much. And I was telling Collins that I gave him 3 lines, and he decided to go and add some things, and he realized getting to the end, he was struggling. He went beyond the script. Thank you very much, and thanks to the organizers. Somehow, despite my protestations, they still got me to come and stand here, because given 45 years, and some of us have been here for quite a long time, and all that we've done, I thought this should actually have been given by one of the younger ones, but KK2 insists that he still wants to put me here, so he has done that. But I'll try and give you an overview of what I've seen the Institute done over 30 years. I wasn't here at the beginning, so 45 years. Some of the years are beyond me. But what they've done in malaria since I've been here. As the chair mentioned, when we joined, it was malaria, nutrition, diarrhea studies, and a few other dotted things here and there. So most of us were working on malaria. And it was what it had to be because malaria was and still a very big problem for the country and at least for our sub-region as a whole. The task is huge, so I'll give my apologies right from the beginning that if I was not able to cover some of the programs that others have done, later on please insert it into the slides or into the lecture so that when this is curated, it will be in. But I've tried to give an overview from the time that I joined till now, what is going on and how it's impacting the current work of the National Malaria Elimination Program and also other studies which hold potential to actually even further impact the work of the elimination program. So let's see what we can do. Okay. So before I go on, I'll beg your indulgence for us to remember some of our colleagues whose work should be here, or who should have been here, and whose work contributed to the things that I'm going to say. I've tried to list the major players that I met and I've been working with over the years in no particular order. And as I said, there are some of our colleagues who have played an important role in some of the studies that I'll describe but are no longer with us. And I'll request your indulgence to observe a minute of silence in their memory. So if we may kindly ask you to rise up and we observe a memory of silence. So may the souls of Emeritus Professor Francis Nkrumah, Professors Maxwell Apau, Michael Wilson, and Dr. Samuel Dinyo rest in peace and rise in glory. Thank you. So the research portfolio at Noguchi is big, it's huge, but I have tried to present studies across these 5 broad areas. And I also decided to present a little bit of capacity building that the studies endanger so that we will have an overall view. As I said at the beginning, apologies for areas that trying to do such a big tour definitely will probably fall through the cracks. So I'll look at work impacting the parasite, the host, the environment, and the vector in what we call malaria. It is a major research focus, and rightly so, because of the huge burden of malaria on the population. And this is in response to the Institute's mandate, which engenders us to research into diseases of public health importance, as well as provide high-end, lab backup to the control programs, and also to provide training. And malaria is one of the biggest health challenges facing the country, as the data shows. It remains a major public health problem, both globally and in Ghana. And it's estimated that in about 4 or 5 years ago, there were about 600,000 deaths, with most of it actually occurring in the endemic countries of sub-Saharan Africa. The WHO has gone ahead to classify this as high burden, high impact countries. Basically, where if we actually put in a lot of effort, we can get some good impact. And Ghana is part of that. And those high burden, high impact countries contribute about 2/3 of global malaria burden in 2020. Ghana's contribution was 2.1%. You may think it's small, but I don't think our population is 2.1% of the world population. So we should really do something about that statistic, and it's one of the things that the Institute has tried over the years to do in support of the previously or formerly the National Malaria Control Program, and now the National Malaria Elimination Program. So despite what has been said about this, over the years, the control program and now the elimination program has done something, and the situation is improving. The transmission is generally stable with varying endemicity across the regions. So you will see in 2011, okay, on the extreme left, 2011, parasite prevalence was relatively high in the northern regions. And over the years, you see it coming down. And these are data taken from the MIX, DHS over the years. And the latest in 2022 shows quite a remarkable decline, with a general prevalence running around sometimes 3%, 2% in the urban areas, and up to about 27% in OT region, which is the highest. 15% in OT region, which is the highest. The epidemiological studies that the Institute has been involved in from its inception showed, this was the early 90s studies when I joined, that malaria was perennial all across the country, mostly the forest and coastal zones with significant seasonal variation. Parasite prevalence was higher in the forest zone compared with the coastal zones, and the sporozoid rate was also higher in the forest zones. And more than 90-95% of infections were due to Plasmodium falciparum, the most lethal species of the infecting parasites. It hasn't changed much because later on I'll show you the current data, which still shows that a large proportion of cases is still due to Plasmodium falciparum. So this is data of some of the work that we did earlier on, showing the prevalence of parastemia and fever, and then severe anemia and age groups. And this showed that most of the infections is in children, which bore the highest burden. It's seasonal in the savanna zone, and the prevalence was higher among children, including those less than 6 months at that time. This was like 1993, 1994 studies that we did in Dodua and Pram, and also in Navrongo at the time. We were able to confirm that malaria was mainly a problem of children. And so it was no wonder that children were the focus of interventions such as deployment of insecticide-treated nets, which was shown in one of the studies, the trials in Navrongo by Fred Binker and others, that they were able to reduce child mortality by up to almost 20%. But then one of the studies showed that there began to the differential response to chloroquine sensitivity by the parasite. And as one moved inland from the coast to the savanna areas, the sensitivity was increasing. So you had a reduced sensitivity in the coastal areas, which probably was driven by drug pressure. So these were the early stages of what we'll later see as was later presented to by the clinicians as go-slow fever and things. And it was around this time that I joined the institute, and we decided to find out how this was actually playing across the country. So we set up around that time the WHO TDR set up the MIM or malaria multilateral initiative in malaria studies, or consortium, and the aim was that they wanted scientists in endemic countries to lead to find solutions to problems of the continent. So malaria, TB, and others, we were asked to come in with our ideas and things, and we will be supported directly. And to a large extent, this propelled the institute onto higher grounds or higher the research areas, because when the announcement was made, the then director, the late Professor Koram, called all of us and said, well, we should be able to respond to this because we've been working in malaria. When I joined, there were studies studying the distribution, the prevalence and things, or the basic epidemiology of malaria in a coastal area and a forest area. That time you could see a clear distinction between Pram, which was on the coast, and Dodua, which sort of had a forest area. And looking at all the parameters from the parasite to the vectors and to how the children responded. So based on that, he assembled us and said we should put in our response. And I was one of the young people who had come back. I had a PhD from London School. My work was on malaria, but it wasn't really on what had been advertised. But having done a PhD, you should be able to respond to these things. What I like about that was how he actually took an interest in what we were doing. And it's not these days of emails and WhatsApp where you can send your document instantly and get a response the next morning or the same thing. So I remember we would come to work on Sundays after church, and we'll present our drafts to him, and he will critique, we'll make changes, he will meet us during the week, and we did that till the time came up and we submitted the application. So I made an application to look at this issue of go-slow fever across the country, because then most of the reports had come from doctors working in Accra and a couple of other urban areas. So I had spoken with the malaria control program people and said, I think we should look at this nationally. So they agreed, and we selected an initial, I think, 6 sites across the country to look at the response of the parasite to chloroquine treatment. This was approved, or this was accepted, and we got a grant. The other grant we got was in immunology, under Professor Diki Akamori and others in immunology, to look at immunological responses in Dodua, continuing the work on Dodua and looking at how the children responded to malaria. The beauty about the thing was, or the important thing I want to point out was that due to the director's actual interest and directing us and actually leading what we had to do, and we also being interested in putting things on, there were 15 awards across Africa, and Noguchi got 2. So then everybody began to ask, where is this institute? What is happening here? But I think, as they say, the rest is history. We've continued into higher studies, into bigger grants and things, and we have a very good malaria program that we all can be very proud of. These are the, later on, the sentinel sites were increased to 10 to represent the then 10 regions of the country. Because when we presented the initial response from the 6 sites, the other regional directors was, why don't I have one in my region? Why don't I have one in my region? And we agreed and increased the size to 10. This was probably, we started in 1998, so this was somewhere probably around 2003, 2005, when we set up sites in each of the regions. These are currently the sites which are still being used, although there are 16 districts at the moment. So I'll give a brief outline on the therapeutic efficacy studies, which I was actively involved. In fact, I started that, and I was actively involved and have been involved up until now. So, basic overview of that. We set up the sentinel sites and then put in a standard protocol to treat and follow up children. We decided to use children because in adults, you have partial immunity. So when you have a partially effective drug, it will appear that things were working well. And that was actually what was happening. The problem of chloroquine resistance was not an issue if you looked at the adult population. So we set up the studies, standard protocol. You treat the children with a standard course of chloroquine over 3 days. This is observed to make sure that they do actually take them. And then we follow them up on a regular basis for sometimes up to a month to look at whether the parasites are cleared, symptoms are gone, and what happened and things like that. To cut a long story short, we realized immediately that chloroquine was very, very ineffective in treating malaria, especially in children. And by 2003, as you see in the top chart there, only about 1 in 3 children were responding adequately to treatment. This was a very important observation because other studies in other parts of the sub-region, I think Senegal, DR, Congo, and other places have shown that the reduction in sensitivity by the parasite actually contributed to increased mortality, child mortality in this area. I think Jean-Francois Trapp and others in Senegal had shown conclusively that when you had chloroquine resistance, child mortality was a problem or increased because most of the problems of the children was due to malaria. So given the situation of malaria, mostly in rural areas where access to health care was not optimum, it was essential that the few contacts that the children made where they should be treated with more efficacious drugs. I have a personal experience of this because in my younger days, I was working as a physician in Boku Hospital. Boku, for those who don't know, is in the northeastern corner of the country. Right now, you can get there in a day. When I was working there, it was 2 or 3 days travel to get there. And the thing was that the children in the outlying districts, we had one hospital, Boku Hospital. Right now, I think the district has been divided into 6 different districts or something. We had just Boku district in the northeastern corner, and it's heavily populated, and the older ones will remember the Unigov days when results will come, and it will come from the farthest part of the country, Boku East, and they had the large numbers and things. Nobody knows how they could get the voice here when we couldn't get into Accra. Anyway, we had one big hospital, or the district hospital in Boku, and people could only access, or most people could access the hospital on market days, which was once every 4th or every 3rd day. So I knew that if a child came to the hospital with malaria, and you were treating the child with chloroquine, which was less than 50% efficacious, and asked the child to go because he was not sick enough to be admitted, you didn't have a bed, or to keep them. In fact, they will not even agree to be admitted because it was a whole different ball game to try and admit one child or admit a patient. You have to uproot a whole family to come from the village and all that. But if in that situation you are using a drug which was less than 50% efficacious, you are probably just condemning 50% of your children to go back and die because you are not really treated them. So for me, it was more on a personal basis, and we felt that we should immediately do something about this. So we went ahead and did a comparative trial of chloroquine, sulfadoxine, pyrimethamine, and then artesunate amodiaquine combination and artemether lumefantrine combination in, I think, 3 different sites of the country for, I think, 2003 and 2004. And the result is what you see in the lower graph there, which showed clearly that, in fact, by the time we did the study, chloroquine sensitivity had fallen down to about 25%. So it was actually at a critical level then for us. So it showed clearly that the ACTs, artesunate amodiaquine and artemether lumefantrine, were good and were more efficacious for us to use. What we actually, in fact, at the time we were going to do this study, we were asked to use AL, artemether lumefantrine, Coartem. But we took a decision that since chloroquine was produced by the local manufacturers, we should put in something that they could also manufacture. And that was the reason why we added artesunate amodiaquine. And we're happy to see that it was as equally effective. So then at least it gave the opportunity for the local manufacturers to continue providing treatment for malaria in the country. Beyond this, we went on and developed the anti-malarial treatment policy for uncomplicated malaria. And I think in 2005, it was officially changed to artesunate amodiaquine and artemether lumefantrine. Later on, it's been updated several times along the line. This is the latest iteration for 2020, where second-line medicine has been added, which is a dihydroartemisinin piperacin DHAP combination. We did not rest there. We've gone on to continue the monitoring for the responses to these drugs, because before we started the chloroquine efficacy studies or therapeutic efficacy studies, the assumption was that chloroquine worked and everybody was using it. So we showed the data, we provided the data to say that it wasn't working. And we thought, well, we can't end there, because knowing the biological situation, the parasites will respond to whatever pressure you create, which is the drug that you give. We had given chloroquine for about 40 years. We're happy, but slowly and surely, they had also found a way to live, because it's a survival. They can't just be swept away like that. So we know that it's only a matter of time for them to respond to the new pressure that we are creating. So this time round, we set up we use the sites to continue monitoring the responses, and this has gone on from 2005 up until now. So almost 20 years that we've been providing service to the malaria control program. And these are some of the results for the efficacy studies that we've been seeing for amodiaquine, artesunate amodiaquine, and then Coartem, and lately for DHAP since 2020 when it was added as a second line. This is continuing, and I'm glad to tell you that this has been expanded recently to add other parts of the story or the other parts of the puzzle to the work. And this will include looking at pharmacodynamics, pharmacokinetics in the children, and I think we've got support to do that for the next 5 years or something. So that is what we've done for that. And on the basis of that, and also looking at the drive towards elimination, we've leveraged the sentinel site idea and actually increased the sentinel size for another set of ideas to look at the parasites, the relative abundance of the parasite species. Because as we drive them down, again, from the biology and from the science, we know that there will be competition and things, and as you drive the things, nature abhors vacuum. Maybe the space is likely to be taken up by the lesser known parasites or the less abundant parasites. So P. ovale, P. malaria, and others. So we've set up sites across 30 different district hospitals where we're using the data to look at the prevalence of parasitemia in febrile cases that appear in the hospital. So you do this on a routine basis or regular basis, and you get some idea of what is going on. And this has been done for the past 10 years or so, and this is what you see for P. falciparum, P. malaria, P. ovale, and mixed infections and things. At the moment, it's still largely a problem for P. falciparum. But we know that to be able to go to elimination completely, when the cases of ovale and malaria and others come in, because of the life cycle and the fact that they go into resting forms in the liver, we need other forms of treatment like primaquine and others to be able to clear the liver forms before we can talk about complete elimination. So this is important, and this is still being done for this. This is what I was saying. So the clearance of the hypnozoids will require primaquine or things. So it's up to us to, it's important that we continue to have a look constantly on this to make sure that we get that. And this is being done between the EPID and the parasitology department in the institute and the control elimination program. So one of the things that has allowed us to be able to show, since to help the malaria control program to know how their interventions are working across the country. Well, we have 30 different places across the country, and we're having annual parasite prevalence rates. They get the survey data once every 4 years, once every 5 years from the demographic and health service, from the mix and things. And it's interesting to show that the rate of decline from the parasite positivity, slide positivity rates and the survey data almost coincide. From 2014 till now, you see this gradual decline, and the gradient is almost the same. So it's a good thing for the control program to know that, yes, what they're doing is working well, and this is continuing to provide data also in support of their activities. The other one that we've been able to do is to test out ideas around mass treatment. And the first time we did this, as was mentioned, was led by the chairman himself in a village in Volta region, where we treated all the children under 15 with, I think we used artesunate amodiaquine. And the assumption was that we're clearing the parasites for whatever it was worth 3 times a year and see what happens. So at baseline, this was a parasite prevalence around 25%. We gave them 3 treatments in the year. Every 4 months, we just gave everybody full course of artesunate amodiaquine. And then came back after 1 year and tested for the parasite prevalence. And even after 2 years, we still have very low rates. So we presented this data to the malaria control program and said, look, because at that time, the debate was whether you should do this because you engender resistance and things. But some of the arguments that we made was that if you look at all the other parasitological diseases, people are actually going after the parasite. It was only in malaria that people were a bit queasy and said, no, you shouldn't do this, you should. But somewhere along the line, this has become now the things that is being done. It started with the seasonal transmission areas, seasonal chemotherapy, which is now a program, activity or a program strategy. But we've continued to work in that area to show that even in the high transmission areas, this can still make an impact. The second chart shows a summary of results from Pakro, which is near Ansah, which has a heavy parasite prevalence, and also just within a year of treatment. So basically, what we gathered from this and we've discussed with the malaria control program is that we need to understand the epidemiology of the areas and actually tailor the response to the places. It's becoming very critical, or to become critical as we move towards elimination, because what we should always understand and know is that malaria is actually a focal disease. The focal disease, because it's driven by the parasite, it's driven by the vector and the activity of the host. So this is not going to coincide uniformly across a large space like Ghana. And if we don't take that into account, we'll be beating around the bush for most of the activities that we do. It's become clear now that as the prevalence rates and things have gone down, this sort of division or this sort of graduation has to be done, and a control program has taken that up. And I think now they have high transmission area, medium transmission areas and things, and they are tailoring the interventions across these things. So over the years, the epidemiological studies that we have been doing has fed into the strategies developed by the control program for what they do. And there's been a feedback loop. They tell us what they find, we put it onto the field, and we go back and discuss with them what is going on. Yeah. So another area that has direct impact in what they have been doing is the entomological studies. The entomology department or unit in the parasitology department has worked extensively and continues to work extensively with the malaria control program. One, because currently the intervention programs depend largely on chemical control of the vector. And this is bed nets or insecticide residual spraying, IRS. And again, we know that in a biological system like this, when we increase the pressure, there are going to be changes. And you need to be ahead of the changes, otherwise your whole effect doesn't work. Or the effect you expect, you don't see that. And this has been one of the strongest areas that the parasitology department has been working on. So they have countrywide insecticide resistant monitoring also across 30 sentinel sites. Initially, these were different, but you remember I presented the 30 sentinel sites for parasite prevalence. So over the past few years, we've actually had discussions, and I think to a large extent, we've merged these sites so that you can then integrate the data and see very clearly what is going on, working with the malaria control people, or malaria elimination people. But basically, most of it is to support prompt decision-making on choices of insecticides, because they have to rotate because the vector develops resistance, and if you're not aware and you continue, you realize that you'll be spraying, but nothing was happening or nothing will be happening. And the spraying is not cheap, so you need to be aware of that. They've also done net durability studies. So the insecticide nets, they check how long they can have the effect over the years. They have standard assays that they do for that. This provides valuable information regarding the performance of the long-lasting insecticide nets. So it gives them an idea of how long people can use them and when they have to be changed and things like that for use during the mass campaigns. They've also been looking at larviciding across some 14 sites to determine the impact of larviciding as a supplementary control or elimination tool and to help manage insecticide resistance. They have the malaria vector surveillance across 20 sites. And recently, due to our trading, activities and things, there's been introduction of Anopheles stevensii. Anopheles gambiae is mostly a rural, dwelling vector. And you see it more often in the rural areas, and that's why we have malaria mostly in the rural areas. Stevensii is adapted to urban areas, so this is something that we need to have a look at. And if you look at what has happened over the years, our urban areas have become less and less susceptible, or have become less and less problematic for malaria. So we are building up more and more of susceptible population for malaria, and very soon even adults will be like children in the old days. And if we are not careful, and within that mix we get a vector which is adapted to the urban space, we may have a problem. So it's very important that this is checked or this is monitored and action taken. And the parasitology, the entomology unit is working on that with the elimination program. As part of that, the vector is introduced from used ties and things which are imported from the Asian countries where stevensii is endemic. So they've worked with the port authorities and things and trained them on how to identify this and also how to control it at the port of entry, and hopefully we may be able to get a grip on this. This shows the susceptibility mapping of Anopheles gambiae to pyrethroids. Pyrethroids are the main insecticides used in the long-lasting nets that are used across the country. Basically, what you see is mostly the red dots, which indicates confirmed resistance across most sites in the country. So we do have a problem with what's one of our tools that we're using. But they haven't stopped there. This is also to another set of insecticides, organophosphates and carbamates, which are used in the IRS programs. But they haven't stopped there. They've gone on to help develop other newer insecticides which can be used both in the nets and in the spraying programs. And this shows what I was talking about, the reduced effectiveness of insecticide classes over the years and the need for new tools which they've continued to work on. So from the entomology studies, the species composition for the vectors were Anopheles gambiae, Anopheles funestus. We begin to see other species, Nili, Rufipis, Kustani, becoming more prevalent. And so there's a need to start screening them for sporozoites to see their importance in transmission. The residual efficacy of current IRS insecticides is generally good, but we need to have a look and see where we can get better ones. And actually new ones are being introduced. VT500 was recently launched. The high resistance to pyrethroids and carbamates in some sites. So one of the big cases or big questions have always been, what is the impact of agricultural use of the insecticides? Because we use the same insecticides for public health as is used in agriculture. And in agric, they want to kill the weeds, they want to kill the weevils, and they just spray. Nobody goes back to find out whether this is staying on or not. And the mosquitoes and the weevils are sharing the same space. And then we have to contend with this. So it's an area that we need to have a look at in things. One of the other things which has been becoming important is the mosquitoes are becoming smarter. They come inside the rooms and we have nets all over the place. So they can't even get a meal. And they need a meal to survive. They need a meal to lay their eggs. So they go outside the room and wait for us. I don't think I have that slide here, but we showed clearly in one of the studies in Navrongo that the biting pattern changes when the net use is high. So the usual thing is that Anopheles will bite between 10 p.m. and 4 a.m., 2 a.m. The peak biting is around that. And we saw a clear peak around 4 a.m., 5 a.m. when people wake up. Because you take off the nets and say, okay, we can have a meal. I don't think I have that slide here, but we showed clearly on that study that this is interesting. So it shows the importance of some of the studies that we have to do or we continue to do here. Sometimes people think, why are you studying mosquitoes, for example? And I remember during the time that I was director and there was doomsaw and we needed power and things. And on one of the stations with the questions and things, I said I needed power to keep mosquitoes alive. And one journalist actually followed me up to this place and said, why we should be killing mosquitoes? Why am I keeping them alive? Fortunately, we had the insectary then, so I took her inside the insectary and showed her the whole setup of the insectary and why we do those studies. And I must say that when she understood it, she went and wrote a very nice story about why the power should not be cut, because the mosquitoes can't sweat and they will die. So we have to get the stories out somehow, but it's very important that we do those things. So the younger ones coming, don't worry about it. There's a lot of things that we can do. So that's that. They haven't stopped there, and other people in the parasitology unit are actually looking at newer, completely novel ways of tackling the vector besides the chemicals that we've been using so far. Currently, what we've been doing is the nets, the spraying, either on inside or outside with the larviciding and things. And the mosquitoes are developing resistance. So this is another map showing the pyrethroid resistance that I showed earlier on. So you can see it developing all over the malaria transmission zones in Africa. And so we do have a problem about that. So what are they doing? They've gone back to look at the life cycle of the parasite. So you have the A section occurs in the vector, in the mosquito gut. When the mosquito picks up the gametocytes and it develops in the guts, and then it moves to the thorax and moves to the head, and then injects the sporozoites again, which goes into the liver and things. And currently, to a large extent, most activities have been looking at treating in the humans and also preventing the bites in the mosquito and things. But what about actually looking at the physiological processes going on in the mosquito gut and see if we can have an impact there? Because that's actually a block. If we can have something which the mosquitoes can feed all the same, but then the parasite cannot develop because we've somehow managed to find how they live in the mosquito, that's fine. Maybe to be a nuisance thing, you don't want the bite, but the bite is not going to give us malaria, and we're not going to have a lot of deaths. So other scientists, they are looking at targeting the pathogen within the vector in the mosquito itself. And this is just so that when we do get a block of from the chemicals, we can have some new things to work. And they're looking at what I call the mosquito biome to look at the stages or to target the parasite stages in the mosquito. So a better understanding of the relationship between the vector, parasite, and the symbiont is needed. Their relevance in natural populations, feasibility as a control mechanism, and this is being worked on here in the institute. Another area of work has been what I packed under the molecular studies. These molecular studies will impact all the things I've described before, the epidemiology and the entomology work and things. And we've had that looking at the drug resistance at the molecular level. The epidemiological studies looked at the clinical side and telling the clinicians or telling ourselves this is not working and things. But what is underlying the resistance, and how do we use those tools to be able to stay ahead of the resistance more or less? So again, in the parasitology department, they've had a look at doing this in a more effective way, using molecular surveillance for drug resistance. And to be more cost effective, because you can actually pool several samples into one, and using the new gen sequencing, drug resistance and things, it provides about 99% concordant results with individually tested samples. Up until now, the way we've been doing this is to test individual samples. And this shows a method that has been developed here and is being called the pooling project. They've gone ahead and actually established sites in the country where this can be done. And currently they are testing that alongside using the rapid method to be able to tell us the prevalence of the molecular markers responsible for resistance in a pool thing. So you can bring in 10 samples and can tell us that, okay, we have an issue here. You can bring in 100 samples and says we are free in this place. As the program develops towards elimination, this will become very useful tool that will be used in the country. So basically, this is what, at the stage in which they are now, they've developed a dashboard. You put in your samples and things, and then it can tell you the prevalence that you have at the place. They've also looked at whole genome sequencing of the Plasmodium CSP protein in 2 distinct ecozones. CSP protein is becoming important because that is the basis of the current malaria vaccines that is available. So they've gone ahead and dissected how the changes in this drives the responses that we see with the vaccine design and vaccine efficacy. Because we know that the efficacy is not uniform across all the sites that has been tested it. And they are looking at this and looking at the reasons for that and see how best that can be surpassed. Another area that has been of interest to me personally is to look at the parasite in detail, what they called complexity of infection, because when we look on the slide, we see one parasite, but at the genomic level appears from the sexual combination reproduction, several levels of parasites with different genetic markers do present to the immune system, to the host, and various responses. And as we talk about elimination, what exactly are we going to look at? Is it that we don't see a parasite, or we've cleared all the various forms at the molecular level, at the genomic level? We've tested some of these ideas in Bongo and looked at, as the control program is putting on the interventions, we've gone ahead and checked the complexity as the intervention works, and seen that you do reduce the complexity from, let's say, 3 or something to somewhere around 1.8 with improved activity. But basically, further work has shown that at that level, there appears to be a block, and we need to find ways of getting rid of several of those genetic variations, the genetic variants. Otherwise, they will come back to populate the same space from the work that we've done in Bongo. The other work that has been done in both the PID and parasitology is to look at the mutations underlying the resistance that we've seen. So we've looked at resistance to chloroquine, resistance to pyrimethamine, all the common drugs that we use for treatment, to sulfadoxine, artemisinin, lumefantrine, piperacin. In fact, these are very important because these are the new drugs that we're using, and we need to be aware how they are. Interestingly, when we've done that, we've seen that there appears to be some reduction in the sensitivity to the ACTs, because the wild type appears to be going down as we've increased the use of ACTs. That's what you see in that chart there. And these PFK13 mutations are linked to partial artemisinin resistance in Africa, and they are in the red color. We've seen that in Accra, in Cape Coast, Sunyani, and Hohoi, in Wa, Cape Coast, and other places, which will sort of give us a little bit of a fright, because this is what we have now, and already it's not half of 40 years. We had chloroquine for about 40 years before we had a problem. Maybe chloroquine problem started in this manner, but we never looked until 40 years. But at least we're looking now, so we're trying to make responses now. But unfortunately, what we find is that despite this prevalence of the mutations, there's a background response to the drug even when the mutations are available. So we are still able to use the drug and things. So this is just a chart showing the various mutations for the various drugs that we use. The 4 things here shows the genetic haplotypes of drug resistance for chloroquine, amodiaquine, lumefantrine, sulfadoxine, and pyrimethamine for C and D. And then FIG4 is the one for the changes that are linked to DHAP resistance. So we have this program, and we're able to talk to the control the elimination program as to how rapidly these are changing and how we should respond. So hopefully we may not be in that situation with chloroquine and at the end of the 90s, early 2000s, and we'll be able to work well. Another area of work has been looking at what happens when we use the rapid diagnostic test. Because the rapid diagnostic test is based on finding the presence of PFH-RP2 genes in the parasite. And already we know there are mutations. It's interesting that, I mean, when you look at all these things, question what I see all the time is that the parasite has to survive. So if you want to find me, I can also dodge and things like that. And it's been shown that when the gene deletions is, so the parasite deletes that gene, you want to look for it using a particular marker, and the marker is not there. So you can't find the parasite, and you say that there's no parasite. But then the parasite is actually there. Again, it has implications for our strategies for elimination, because if we're going to find it and we don't find it, we'll still get people being sick and things. And it's been shown that from the data that when you have the deletion is more than, say, 5% in the population, you have a problem in terms of getting a lot more of false negative results, and it affects your program. So we've been working with the elimination program to look at this across all the regions. Fortunately for us, we're still less than 5%, so we can use the things and work. It will still work efficaciously. Then I would give a brief overview of immunology. I can say that I've worked with them for all the time I've been here, but I still don't understand most of the things they do. Dicky never never put me into the things. But basically, I do get a bit of it and things. But I tried to look at that things. So in the department, my colleagues are taking an integrated approach to studying malaria transmission. We've been talking about treating the parasite in the human, looking at the parasite and the vector, looking at the vector and things. But one of the bottlenecks is actually the change from the asexual reproduction to sexual reproduction. And if we can nail that, then we can actually stop the transmission. So they've been looking at gametocyte biology and looking at the diversity which is associated in variations in the for developing transmission blocking immunity. And also they have systems looking at drugs or things which can affect the sexual forms, which, as I said, if we can knock it off, again, the parasite, the vector may bite us, but there's nothing to pick, and we can actually stop the transmission. They have done that, and they're looking at ways in which this transmission is affected, and together with the secretary, the Vestergaard place, they have developed feeding plans, assays and things to test all the various things that they're doing. So the immunology, I'm trying to, with the help of my friend and my colleague KK, to capture most of the things that they do here. So let's see. They're trying to look at peptide discovery. This allows them to look at the parts of the parasites that the host responds to. So identifying targets for vaccines against liver stage and overcoming human gene diversity. We know that the parasite also gets into the system. In fact, if you look at the life cycle, when you get bitten by the mosquito, nobody knows how long the sporozoa stays from, let's say, a bite in your arm and getting to the liver. It's been estimated from seconds to minutes to days. But we know that within some time, it will disappear and get into the liver. And when it gets into the liver, is more or less lost to the body. Your system, which appeared and saw something from the parasite, which is the sporozoids, and started producing antibodies to that. Now has produced antibodies. He comes and there's no sporozoids. They've all disappeared. They are in the liver. So the parasite is very wily, and we have to do a lot of work. So they're looking at how the parasite have this capacity to escape the immune system, and they're formulating vaccines with different variants of the same antigen, for example, and to prevent the different forms of the parasite from hiding in the immune system. They've also looked at antibody targets of protective immunity. This is an area that has interested me a lot, because in my training in Gambia and when I was working on severe malaria in children, you will go to an area and you take 2 samples, or you have 2 brothers or 2 friends in the same vicinity, sometimes in the same house. They're about the same age, and one child is playing football, the other one is comatose in the hospital bed, and you wonder what is going on. The responses are completely different, and we need to understand this and be able to help, because the comatose child, if you are not careful and we are not quick enough, we may lose within 24, 48 hours, for example. And this is quite an interesting area that we've been working on since I joined with the Copenhagen group in Dodoa, Dickey, and the others looking at this. And they're trying to identify the targets for vaccines against blood stage. But mostly is work on the blood stage things. The acting director has been working mostly on malaria in pregnancy. Again, it's known that the pregnant woman is susceptible to malaria, especially the first pregnancy. And subsequent pregnancies, somehow they build immunity to it, to the extent by the 3rd, by the 4th pregnancies in the same area, nothing really happens to the fetus and to the mother. Again, if we can dissect out what these changes are, we'll be able to have some vaccines or some protection for the unborn baby. And in fact, work is going on with our colleagues in Denmark and other places to develop a parasite-specific vaccine, the VAR2 CSA development, which I think has been tested in Benin and other places. The ultimate goal is to have a product in a bottle which we can use for especially our younger sisters, mothers, when on their first pregnancy, so that they can go through that without much of a problem. I've mentioned transmission blocking vaccines, which has become an area of interest, and I'm hoping that we'll be able to have a product which we can then test at the clinical trials unit to see that, yes, when you get this, you don't produce gametocytes, or your gametocytes, the gametocytes when they are produced in your body are not infectious to mosquitoes, and we can go ahead and use that on the population. Lastly, they're looking at antigen production and looking at getting proteins that we can use in our studies and also as basis for vaccine trial. There's a whole lot of other things which are going on. As I said at the beginning, I might have missed some of them. I know between clinical pathology and the animals units, there's been a lot of testing of our traditional plants and things on efficacy for, as in the efficacy for malaria, but I've not been able to present that because I haven't had a lot of work, a lot of input from that end. So I think this is the end of the tour that I was able to develop. And basically I was saying that I had a few slides, a couple of slides for capacity building, which I think is quite important for us to indulge in. There have been several opportunities to train younger ones, but what I brought, what I'm interested in is the activity of the entomology unit actually helping the control program or the elimination program to monitor the vectors in the field and also at the entry places. So that's what I highlight in this talk with the training given by the entomologists to the field technicians in the measuring of vector sensitivity to insecticides. Earlier on when I joined, there was the mid-level Ministry of Health, Ghana Health Service technical training, where actually people spent up to about a month here doing microscopy and other things for malaria and other areas, parasites, which have direct bearing on the work of the control programs. And so some of the pictures from the entomology field work, sampling for larvae, sampling for the vectors itself and back to the lab, and also the port surveillance for Anopheles Stevens High, which has been going on at various staff places. So, in conclusion, the Institute has, over the years, conducted several studies in direct response to its mandate on working on diseases of public health importance, and malaria being one of the major problems of public health in the country. We've worked on that in several areas, from epidemiology, entomology, molecular biology, immunology. We've also had studies on ethnographic studies. In fact, it will be interesting to repeat those ethnographic studies now, 20, 30 years on, when malaria is less of a problem. And there's been a lot of education on the public space to see whether there's been a shift in those ideas that we found at the beginning and how we put this into the current drive for elimination. The research has spanned, basic science to applied research, so from the bench to the field. We've looked at the basic science as a potential to affect future strategic directions of the elimination program, molecular markers of parasite resistance, for example, vaccine research, transmission blocking studies, new drugs, new vaccines, and the direct or applied research areas that we continue to do, which includes the therapeutic efficacy studies, the entomological monitoring studies for the vector competence, vector susceptibility, and also the diagnostic testing we're using the RDTs, are all things that we've been doing over the years to help the National Malaria Elimination Program. So this will continue to generate, we'll use the Sentinel sites to generate the data, the entomology, and other things. And I was mentioning that we've had support to expand the therapeutic efficacy studies to look at both in vivo results, molecular studies, and look at pharmacokinetics, so that we will be able to talk, to say that the drugs are being absorbed well and what the level of the absorbance on what we see at the clinic and all that. We've had support from several areas, and I'll manage to pick this up from the slides I got from colleagues and the acknowledgement that they did. But I'm sure there are others that I probably have missed, and apologies for that. But basically, we've had support to look at all these things from several areas, several funding areas, and we are quite grateful. Thank you very much.
I think we can do it better. Let's standing here for an hour. Thank you. Thank you very much, Professor Koram, for educating us on malaria work at the Institute from all angles. And I must say that malaria is one of the disease areas that have received a lot of attention at the Institute, and you clearly demonstrated that to us this morning. So now the floor is open for questions. And so you can sit down and take the questions. Right, so.
Yeah, thanks very much, Prof. Koram, for enlightening us on this 4+ decades of the work that has been going on here. Towards the end, I was preparing to actually ask a question regarding the role of medicinal plants, because I know that a lot had been done, but you actually just gave a comment on that. Can you throw a bit more light? Probably others may throw a bit more light on what has happened. I love taking Ibima a lot because I've found that to be able to help me more than the other drugs. So just wondering. Then the second is just looking into the future. With all this galamsey, and we saw a picture, and the two of us were wondering whether that was taken from a galamsey site. So looking ahead, what is going to be the impact of all these things going on in the galamsey areas, and these are in rivers, of course? Eventually, there is no doubt that this will impact our malaria control or the management of malaria in this country. How are we preparing towards that?
Good afternoon, everyone. Or good morning, it's morning. Good morning, everyone. Prof, thank you very much for your very insightful presentation, and also I do appreciate the kind of work you've done over the years. Congratulations on that. Mine is on your earlier studies that you did. The one you looked at chloroquine, artesunate amodiaquine, and lumefantrine. I realized that for chloroquine, the sensitivity was very low. So, I mean, it stands to reason why you advocated that we should stop using it. But then if you look at Koram, that is artesunate lumefantrine, the sensitivity was somehow around 97.5%. But we are still heavily reliant on that medication. Why is it that you don't go ahead to advocate that now we should strictly be on artesunate instead of still using Koram? Thank you.
Thank you, Prof. Koram, for the presentation. So my question is on the use of MDAs. So there's been this argument about its impact on resistance and whether we should focus on using it to reduce malaria transmission in areas of high burden, as we do with the SMC, and then also recently expanding to school children as well, or whether we should focus MDA on areas with low transmission so as to just eliminate malaria in those areas. So I want to count, based on your experience, count your thoughts on that. Thank you.
I think let's allow Prof. Koram to respond to these questions, then we can take another set. So, Prof.
Okay, I will try, but I have to pass some of the questions on. On the role of traditional medicines, traditional plants, as I mentioned, in trying to get the overview, I didn't have much. I could have put a lot of some of the things in there. There's been a lot of work, and maybe Regina will want to fill in some of these things that you've done at your place, because I remember some of the early studies for your masters and things which we supported was looking at some of the plans for the treatment of malaria. Galamsey is a very complex issue. And if you look at it, I think some will have to correct me, but if you look at it in respect of the vectors, I think Galamsey will do away with the vectors. Because Anopheles is a very neat vector. The mosquitoes, they don't like all that dirt in here that you are creating. So on the vector side, probably will say that the Galamsey will do away with the vectors. But it will not also be that simple, because when they are doing the activities, that's when it is. But when they move on and the stability and things settles, they will become huge breeding grounds, as I suspect it will be. And that will be another, as I said, it will be a complex thing that they will have to do. Looking at it, we'll have to regulate the thing properly and make sure that if you dig, you fill up. Because if you leave it over time, they will become very big pools and things, which will become, breeding grounds, which we may not be able to control very well. The other one I'm afraid of is what is being done to the host itself, because we are taking in all sorts of things, and maybe my immunology people will tell us that, with all the chemicals, the heavy metal poisoning and things like that, our immune responses may go haywire or whatever, and we may not be able to respond properly. The one on the, I think it's Kofi Boni, on the comparative studies, I think you have to get the thing right. We used artesunate amodiaquine and artemethyl lumefantrine, which is the same as coartem. Yes. And I did not present the confidence intervals on those charts, but 97.5% and 100% probably about the same things. I try as much as possible not to throw these words out, especially during lectures and things like that. What exactly do you mean by significant? Are we speaking English or we are talking stats? Stats. Pardon? No, but we did a one-off comparative study in 2003 to 2004, thereabouts. If you remember one of the graphs that we showed, which they've continued to look at this, they approach 100%, but not all of them are not 100%. I think Ben may be able to expand on that. You will get variations on the thing. Life is not a flat one-line curve. You know it. Your viruses, did they all respond the same? No. So please, don't read too much into those things. And when you look at those things, as I was telling you, we intentionally included artesunate amodiaquine because that would give a room for local manufacturers. At the time, what was being promoted was coartem, which we found to be at 97.5%. I think subsequent years we found it around 100%, 98% and things. So there's variation into those things. And then MDAs and impact on resistance. Again, another area of interest to me. If you look at the other parasitological, the parasite diseases, there was it, NTDs and things. Everybody has aligned a rail of drugs to go and knock out the parasite. Why is malaria different? Why don't you want to knock it out? I still haven't gotten an answer to this. But again, if you look at the epidemiology, for example, a place in Navrongo or in the Savannah Belt, the transmission is heavily during the rainy season, May, June, when it starts raining, till about now, October, November. By December, the tail end of it is past and everything. And from December, January, all the way to another May, June, you can hardly get anything. It doesn't mean that they are not there. But there's a bottleneck that if you drive them down, the pool for the next transmission season will be lower. Maybe my mathematical mind is telling me that that's the way to go. Because I've always believed in this. And I remember in, I think, 1995 or something, 1997, we were making this argument in Ifakara. And it was quite interesting to see us with some charts and things with Kilama and other people. And people thought, well, this will work. But then they brought up this idea of resistance. But it was, I think, Prof. Binka, who said, why are we enamored with this resistance idea? Let's do it and see what happens. After all, you guys do the molecular studies and things, and we'll respond to it. So frankly, I will always advocate for MDAs. And the thing is that you need to understand where you are. As we showed clearly in a slightly lower transmission area on the coast, we're able to have a significant effect with a long-term things. When you come to Pakro or something, you need to have a different strategy. You may have to treat every other month for maybe the first 2 years and things before you go down, because it's work that we will have to do, and we'll have to do it well. I don't think resistance and things should really bother us.
Thank you. There's a hands up here, and Nancy. Okay. There's Prof.
Thank you. So a little response to, yes, thank you very much. The Department of Clinical Pathology has been doing work on anti-malarial drug discovery over the years. And there was no request for slides. I would have done that. Reverend, are you listening? Okay. So we have done quite a bit of work, and we have found very interesting agents. If you look based on what Rachel says about extracts that are effective, we have found very interesting agents. And so I think you may want to read more on what we have done. There's very little time to talk a bit of it, but there's a lot of work that we have done in this area. Quite recently, we have published a paper also that has very interesting findings you may want to look at.
The mic is given to Professor Nasi. I saw her hands up. Okay.
Yeah, thank you, Prof. So this is just a disclaimer for our media brothers and sisters. Noguchi has not said that because of the Galamsey dirty water, the mosquitoes are not breeding, so malaria will go down if Galamsey goes up. Thank you.
Yeah. Thank you very much. Thank you very much, Koram. That's an excellent summary of what we've been doing in malaria, as you said, since you came to the institute. But I just wanted to add a couple of things that, I think this institute has really spearheaded malaria research right from the beginning. For those who have been associated with the institute long enough, you may recall that the institute was the first to actually convene a national malaria seminar. It took place in the auditorium of the medical school in 1989. Sorry. But you see, it's very important to highlight this, because it brought together all the stakeholders that Kwadwo outlined, who have since ensured that we've gone from basic research to applied research and to policy. And that's what makes the difference for people who are depending upon the findings of research, communities, populations, all of us, that the research ultimately benefits us health-wise. I mean, public health. So I think this is very important. And that meeting actually involved the Ghana Health Services, then of course the Ministry of Health, so much so that it drove all the other activities in malaria at the institute, and which you have heard so much about. And I think this is great. Just to also complement, the answer, the response on plant, research, or to look at natural products. As a component of the Danida project, this is the Danish International Development Assisted, grant which was given to the institute to help enhance research capacity. There was a component which dealt with, looking at natural products, and there was a team from one of the universities in Denmark working with the chemistry department and the Center for Scientific Research into Plant Medicine at Mampong. They did quite, a lot of studies. Some PhDs were trained on this program from the chemistry department. And I think there are a couple of publications. So, Prof, it's something which the institute has been, involved with. And so it's just that, Koram, of course, you have put a huge burden on him to gather all the evidence, and it's not easy. I must say he's done an excellent job. So that's why he couldn't cover all of this. Thank you.
Thank you. Good morning. Thank you, Prof. Koram, for enlightening us once again with this very insightful lecture. I want to find out a few things. The first one has to do with science communication over the years. With respect to your work, how has your relationship with the media been? And to quote or to bring back what the example you gave, where it took an unfortunate national crisis to expose the need for breeding mosquitoes in the whole malaria research area. So how has it been, and what are the challenges, and how do you think it would go on from here? That is one. And two, is there any data as to how self-medication on anti-malarials contributes to the emerging resistance in some of the regimen that are administered, especially in the case where most pharmacies and dispensaries do not really carry out the test before, giving out the anti-malaria. You have a headache, you fever, okay, Quatem will do for you. So how does that contribute to it, and what's the policy towards that direction to involve all these stakeholders in helping the elimination program? Thank you.
Thank you very much, Prof. I wish to find out whether there has been some moments during your tenure when you had a difficulty in getting your research agenda implemented at the level of the Ghana Health Service or the Malaria Control Program. That was my first question. Then secondly, what is your opinion on the SMC implementation? Where, for example, we see that in a household, you will see a parent and the other older siblings being positive for malaria, and then only under 15 or under 5 children are treated, as has been the shift in policy. So, have you had a challenge with this? Thank you.
Okay, so the first one was science communication. May I know your background, please? Biomedical engineering. Do you plan to go into journalism? No. I'm interested because it's been a struggle on this, and I'll tell you what we've been able to do or what we did over some time now. As I used to tell some of the people, we are off the scene now. We should be off the scene now. But we actually formed, what was the name? A malaria group with some journalists to try and actually communicate these things properly and things like that. Unfortunately, the journalism which is being done is still at the sensational level. Unfortunately. So that one of the instances, I think Dickie may remember, there was something about malaria vaccines or something. Somebody wanted to interview me about malaria vaccines and things. So I said I was busy. He said, okay, you come the next day or something. And I remember, I think we were having some meetings down in the seminar room. Called me on the phone. We spoke about some work and things. And I was like, okay, we are not actually doing a malaria vaccine study, but we're doing studies which could lead to malaria vaccine. Apparently, what he was looking for was that he wanted to fill the back page of the following day's graphic with a story. So my conversation with him, trying to explain the work that my colleague Dickie was doing in terms of some of the things I described, came at the back of the graphic the next day. Noguchi is developed malaria vaccine, according to Professor Koram. So that's what I was asking whether you're interested in. It's important, but I think it has to be done from both levels. And I've sat in several, journalistic or media relations and things over the years in terms of trying to look at this and things. And there are several stories that I can tell you. One of the ones that also hit us very badly was when we did the efficacy studies and showed that chloroquine was not working. And then we said we should use artesunate amodiaquine. Amodiaquine is very good in children. Somehow, the way we adults metabolize it is different. Some of us have the clinical pathologies with cytochrome P4 something, 450. Others don't have and things. And if you don't have, you're unable to break down the drug quickly and it builds up and you get very bad side effects. I had a reaction when I was a medical student when I took amodiaquine. I mean, I could hardly walk. And I could hear somebody say that I was drunk. But that's what happened. So when they introduced this, remember we were working with children. And across our study size, the mothers were calling the new drug that we have brought them One Touch. That was when Vodafone and things had come. So the mothers called it One Touch, that if you gave it to the child, the fever disappeared, the disease is gone. So the mothers were happy. They called it One Touch. The adults took it. Some of them reacted. And I was actually called in the papers that we were killing Ghanaians. So that's the bit about sensation. But we need to work at it. And I agree with you. And I don't know whether the training schools and things, we've done some bits about it. I'm trying to remember this thing that we formed with a charity Binka. So, what is it? I'm married for something. Malaria, something. So we're actually trying to give those things, and we try to do that. So relationship with the media, it's interesting. Sometimes it gets very complicated. Data on self-medication. The policy in the country now is test before treatment. Whoever wants to treat has to test. And at least not microscopy, all of us, I think we've stopped using the microscope. We've all forgotten that. But the RDTs, everybody can interpret the RDTs. And I know they have a program of actually making sure that pharmacies and things actually do have RDTs to test before they do treat that. So if it's not happening, it's a bit unfortunate, but that is the policy of the elimination program, that you should test before you treat. Research agenda and the National Malaria Elimination Program. I think the trick about it's one of the things that we used to do when I came in, which we called our annual meetings, annual research meetings. And the annual research meetings was actually for us to present what we've been doing to the national disease control units, the various disease control programs, and also for them to tell us what areas they would like us to investigate on. So if we have that, if you have that going, you don't have a problem about testing your ideas. Some of the new ideas, sometimes I know it may not, immediately be taken up on board and things. So my advice is that they may not be able to support you on some of the things that we want to do scientifically and things. We should find a way of getting that done and then present them the data, which they will buy into. So we've been working with them. In fact, for me, all my time here working with the National Malaria Control Program and our elimination program, I haven't had that much of a problem in terms of what they need and how we're working on that. There are other areas which are of interest to me. The immunology and things like that we've been doing, for example, may not be something that they are interested immediately, but basic science things don't usually go directly to the control program people, so don't pass that to them. Then you were talking about SMC and treatment of adults in the same vicinity. I think with the program moving towards elimination, this may become a moot point because we have to treat everybody. Otherwise, we can't get to zero. So currently, they will have those things inside and those things. But once we're able to present the data to show that it's not only, and everybody knows, it's not only the children who are going to transmit the things. The adults will also contribute to the transmission. So I believe that somewhere very soon, we may come to a point where, if not regularly, maybe at least once or twice a year, we'll try to knock off the parasite in all the populations. And that's the only way we can get to zero. Thank you.
All right, thank you very much. I'm Wisdom from TV3. I'm their head of data research. And I like the comments that has been raised. And I would like to say that we at Media General and TV3, we appreciate Noguchi, and we have put measures in place to ensure accurate reporting as well. Usually, it's about the angle of the news. You see, we have producers, and most of the producers want a punchy angle. So if I go with, hey, Noguchi Galamsey, it's a topical issue, and they are going to pick it. So we appreciate that, but you also have to bear with us, okay? So we've put measures in place. We've put measures in place to ensure accurate reporting. So I heard Prof very well. He was saying when it's about a vector, mainly because there's mercury cyanide in the water, they might kill the vectors. But if it settles, if it's... Okay. So
sorry.
If
I mean, I'll sit down. We all listen to this. Sorry. We all. Excuse me. We all listened to this very excellent lecture, and I think that in response to the question raised, what I recall is Prof. Koram saying that Anopheles mosquitoes like clean, relatively clean water to successfully breed in. And then he also went on to explain that with dirty water and high turbidity. So I think you better keep that in mind. I think he also added that where people dig pits and they actually cover them, so there's no opportunity for water to accumulate, it will definitely prevent mosquitoes from breeding. Those are the things I heard. I don't know whether anybody heard anything else.
Thank you very much.
Please, can I go on, continue?
Yes, please continue.
Okay, so thank you very much. Well noted. And he also raised a point that if they leave craters or they leave holes, maybe when it's stagnant, it will breed mosquitoes. It settles, the turbidity settles. Okay, I have to get the lecture notes after here, though. So we have put measures in place to ensure accurate reporting. And this is my question. My question has to do with, in the course of your research, what are the hotspots you've identified to be a place where the breeding of mosquitoes, or you have identified as hotspots of malaria prevalence? And do they have a common characteristic that you've actually pointed out that across these areas, there are these particular characteristics across all the hotspots you've identified? Thank you. And we'll ensure accurate report.
Yeah, thank you, Prof. Koram. I just want to bring some clarity on the issue of galamsey. In actual fact, when we say turbid water, you say Anopheles prefer clean, water. When water is turbid, it's not polluted. So turbid water, the picture you saw, where you saw some staff collecting larvae. They were collecting larvae from rain water, okay, which is turbid. It becomes brown. It's not polluted. Polluted means you have water that is smelling and has some organic matter. Anopheles normally will not breed in those. They'll breed in water that's very clean, directly exposed to sunlight. Now, what Galam said that, in fact, the past 2-3 years or so, we saw malaria prevalence actually shifted from the northern part to the eastern region. And then just recently, also in the western region. And we have Volta or T region now having the most prevalent. What we found is that the crater that's left with the galamsey, the water there is brownish, is turbid, and provides breeding, a good place for breeding Anopheles mosquitoes. So galamsey, in actual fact, really, in some leads to providing breeding sites for breeding Anopheles mosquitoes. So you see malaria prevalence goes down. In some cases where the water is turbid or very deep wells, you don't find normally mosquitoes breeding in those kind of wells. So it has an impact on malaria in the sense of providing breeding sites for breeding. So that's the clarity I wanted to bring on that. The other thing with the question you asked about hotspots. So you have different species of mosquitoes. They'll breed depending on the ecology, the type of breeding site. Anopheles, once we are talking malaria, I'll focus on the Anopheles mosquitoes. As Prof said, they prefer clear water that is directly exposed to sunlight. So that's why normally malaria transmission comes during the rains. When it rains, you have water. Even the small pockets of water you have just behind your house. When you look in, you find a lot of larvae breeding there. The new species that we found, the Anopheles stephensii, is unique. And that's where, when you talk about hotspots, it's a challenge, because recently one of my students did some modeling on the new species, and using some parameters, temperature, population density, we're able to establish some hotspots where this species is likely to
inhabit. And we are currently monitoring those sites to see whether we can detect the new species in Ghana. So it depends on which area. But of course, mosquitoes, distribution, density are shared back to you. No, but hello. Are you okay with that? Or if there's any clarity, you see me after the meeting.
I think what I would say about your question is this. As you said, in fact, I mentioned that, and that's what I meant by, I have interacted a lot with the media space in terms of what we do and things. But basically, we still haven't had a level of journalism where I would take a paper and there's a column which is talking about, say, malaria. And I will actually use that to learn about malaria. We don't have people like in your space who will take their time and understand, so that the average person who is reading that column will know a lot about malaria in terms of the vectors and things like that. All you will see is Galamsey gives malaria, which, yeah, is news. But I think journalism has to go a lot beyond the news. Because I would like to take a paper and read on somebody who is talking about economics and things, and I will understand and see what these things mean. But I don't find that in any of our papers on the TV stations or anything. You don't. In fact, one of the things which I find very, very difficult to listen to is the discussion program on air. You hardly ever bring anybody who is going to educate me when I listen to what is going on. No, it's a very serious thing. And I've always said this, that I find it difficult that for whatever topic that you want to discuss, you have the same discussants, for example, because they are the media people that you fall upon. Well, maybe that's how you operate and things, but you need to have a look at that and begin to actually use the space to educate people. Well, we all need education. Thank you.
So, before I end, there is an anonymous question here. Prof, the person wants to know whether you are monitoring resistance in collaboration with the neighboring countries.
Yes, we do. We have, one? No, there was one and then something else come on. So we have a group where we look at this and the countries around us, and I think all the sub-countries, all the countries in the sub-region belong to this, and we do meet from time to time to look at the data and see what happens. In some situations, we've intentionally chosen like border areas to look at what is going on and things like that, because we know that, yes, the mosquitoes, they don't recognize any of our borders, so they don't care. Yes.
So one last question from Prof. Sama.
Well, it's not a question. Kwadwo, I think the media group that we're trying to very hard to remember is the African Media and Malaria Research Network. Yes. Yes, and they have all the information you need. Thank you.
Thank you very much. And let's sit down, Prof. Right, so I believe you all agree with me that it has been very insightful, very educative, and then, if nothing at all, you now know that at Noguchi, the malaria work is very broad, starting from epidemiology, social science, immunology, and parasitology, entomology, and even drug discovery with a herbal screening. That is the work taking place here. And the focus going forward in the recent years is how to generate data to support the elimination agenda of the country. And I want to add to what the question that Indon asked. Why do you treat the children and leave the adults? And the adults become the reservoir of the next infection in the children. So all these questions has to be answered. And I always say this. In fact, I made one of the things that Prof presented. I presented it at ASTMH, and the question everybody was asking is resistant. And I said, why is it that when Europe and America wanted to eliminate malaria, they used all the tools available? They gave the drugs. They used the DDT. But when it came to the turn of Africa to do same, they say resistance. And nobody could answer because that was the truth. And everybody said, yeah, you are right. Yeah, you are right. Yeah, you are right. So the question they asked me, I threw it to them, and everybody said they agree with me that they did all the things that they could do to eliminate malaria. But the bottom line is the ball is in our court. We have to make decisions for ourselves. We have to plan our own elimination program and develop our own agenda if we want to eliminate malaria. Nobody will do it for us. They will always have problem with whatever we want to do if we are going to them to ask for money to do it. On that note, I want to bring the curtains to the end and then call on the MC to give us the announcement. And before that, I think you deserve another round of applause.
Thank you, Prof. Chair, Professor Colin Stephen Aholu, for chairing this session. I have a quick question on a lighter note. I heard the new species of Anopheles. It's Anopheles stevensii or stevensii, depending on where you are coming from. Prof. Stephen. Anyway, I'll see you in chambers. Yeah, and
maybe Stevie was the first to discover
that. Yes, and so there's a lot to be done. The young ones coming, don't relent. Prof. Koram indicated that there's still a lot to be done in this area. Now to a few announcements for the day. Our next seminar is on 31st October 2024, and it will be delivered by Dr. Franklin Esiedu Bequin. He is the Director of Public Health, Ghana Health Service. He will be speaking on the topic, the impact of Noguchi on the Ghana Health Service Disease Surveillance and Emergency Response System. Then after that, we'll have the climax of the 45th anniversary in November. On the 7th of November, we will have a panel discussion, we'll have a debate. Please, everybody be here. The feeling when you are here is different when you watch online, so please come and don't miss that session. And then on the 8th of November, we have the open day. Noguchi will open its doors to the public. We'll have a lot of school children coming around. We'll have adults also coming around to see what we have been doing over the years. Now, without any more announcements from the back, I would like to thank you all for coming and have a wonderful day. Thank you. Oh, sorry, we started with a word of prayer. I would like to call on Reverend Mrs. Eunice Ampim Danso to say the closing prayer for us. Thank you.
Thank you very much for having me. Prof, thank you very much for educating us. Shall we please bow our heads for a word of prayer? Heavenly Father, we are grateful unto you for all that you have given unto us this morning. We are grateful for the advancement in research. We celebrate our achievement, and we also acknowledge our challenges, Father, that will foster Father, future discovery, even in the name of Jesus. Right now, I pray and I use all the Father researchers that have been enlisted for this lecture series as a point of contact to all other researchers. We ask that, Father, you will preserve their lives, oh Lord. Daddy, Lord, we need more guidance in research. Daddy, we need our mentors alive. I therefore pray and ask that, Father, the strength of the Lord shall be with them in Jesus' name. We pray that you will increase them, Father, O Lord, in strength, in knowledge, and understanding, even in Jesus' name. Let the right hand of the Lord be upon them. Preserve their lives, even in Jesus' name. Your word declares that you will satisfy us, Father, with long life. Daddy, not only long life alone, but good health, even in Jesus' name. Satisfy them with peace, O Lord, the peace of God that surpasses all understanding, that will enable them, Father, O Lord, to invest their time and energy into more findings. Father, let it be their portion today, even in the name of Jesus. Daddy, Lord, we bless, Father, every researcher over here, and those, Father, who are not even with us. We pray that, Father, you will continue to make research, Father, in this institute and in Ghana, Father, as a shining light, O Lord, that will shine brighter and brighter, Father, to a perfect day, even in Jesus' mighty